Hello, my name is Arna, and I wanted to talk about how myotonic dystrophy type 1 is an autosomal dominant disease that is caused by a repeat in the genetic code. The RNA sequence CUG is repeated 50 to over 1,000 times in the DMPK gene, which causes MBNL1, an RNA splicing protein, to be overused. A way to tackle this is by using a targeted peptide-based drug. It is smaller than a large biologic and yet more specific than a small molecule drug. This allows for easier delivery and less side effects. In our experiment, we hypothesized that we would be able to find a peptide that would specifically bind to the harmful RNA sequence in hopes of modifying it into a drug. The process of identifying a base peptide is through a display screen. The display screen starts from a DNA library which contains the sequence for a 10 random amino acid chain. The library is transcribed into an mRNA sequence which is then ligated onto a puromycin-DNA tag. This is so that once our enzymes are done translating our mRNA into a peptide chain, there is a buffer just long enough for the puromycin tag to attach to the molecule. To stabilize this and ensure that our mRNA doesn't bind to anything else, we add a cDNA strand. This process creates a fusion library containing mRNA, cDNA, DNA, puromycin, and the peptide chain. This peptide acts as a substrate and binds to an RNA target attached to magnetic streptavidin beads for selection. Due to the high variety in possible sequences, repeating this process a few times ensures that we are able to select for the peptide with the highest and most accurate binding. But wait, does this actually work? So far, we were able to select for a peptide that is specifically bound to the RNA for MD1. The sequence for Huntington's, which is a CAG repeat instead of CUG, was used as a negative control to avoid any nonspecific binding. I wanted to thank Dr. Arthur and Dr. Prabhakar at Frederick Community College for providing me with the opportunity to work with Catoctin Bio under Dr. Regan LeBlanc, who mentored our team.